So a quick recap, what we've done so far this quarter. We started by just discovering how can we even assemble the genome sequence of an unknown virus. And then we talked about how can we annotate this genome that we've assembled. And then we talked about like how can we do sequencing in the midst of a pandemic. So we talked about like consensus genome sequencing, stuff like that. And we've talked about, how can we do multiple sequence alignment and phylogenetic inference to make some inferences about like what is the relationship between different genomes that we've collected from different people. So now we're finally able to actually do stuff with the information that we've tried to receive. Now we can actually try to connect it to some type of public health. So today we're going to be talking about how do we study the prevalence of mutations. And the topics that we're going to talk about today, first is this computational topic of ancestral state reconstruction that I'll tell you what that means. Then the next one is called dating of phylogeny, not like dating like social, but dating like putting dates like times on a phylogeny. Then we're going to talk about assigning genomes to lineages. And lastly, we're going to talk about how do we actually study the prevalence of mutations. So first, ancestral state reconstruction. Just a quick recap of what we've learned so far. This is the true evolutionary history. I have some ancestral genome and mutations happen down this genome. We've seen this figure a few times now. And then kind of in practice, we don't get to see the actual phylogeny. We can only see the present day sequences. And then phylogenetic inference, what we talked about last week was how do I estimate the evolutionary history from just the sequences? But this evolutionary tree that I get doesn't have any of the labels on it. It just has the tree structure. So ancestral state reconstruction is this problem of how do I reconstruct the sequences of the internal nodes, right? Phylogenetic inference was just how do I reconstruct the tree structure itself? But now I actually want to figure out what were the sequences of each of these ancestors. Okay, so that's the computational problem. My input is my multiple sequence alignment and my evolutionary tree. And my output is what is the ancestor sequence on each of the ancestral nodes in the tree. So one approach that we can do this is called maximum likelihood ancestral state reconstruction. And the term maximum likelihood should hopefully sound familiar from last week. We talked about maximum likelihood phylogenetic inference. So inferring a tree that maximizes the likelihood. It turns out that we can do something very similar to the scoring algorithm we talked about last week to do this. Remember last week we talked about here's a bunch of different models of evolution. And under those models, we can come up with ways of defining the probability of tree or like all those types of likelihood things. And we wanted to take the tree that maximized the likelihood score. Well, we could do the same type of approach where we can try to do a traversal of the tree. So post-order traversal is a computer science term. If you haven't heard it, that's expected. Actually, you probably wouldn't have heard it yet. But basically, it just means you're traversing over the nodes of the tree such that you've looked at, you look at the current node after you've already looked at its children. So a post-order traversal is I look at myself after I've already looked at my children. And I'm going to show you how that looks like on the tree. Yeah, so basically, we traverse over all the nodes in the tree, kind of bottom up, starting from the leaves and then going up the tree. And if the current node that I'm looking at is a leaf, I already know its sequence. That was given to me. That was one of the inputs. Otherwise, so if the node that I'm looking at is an internal node, so a node kind of ancestral in the tree, for each position of its genome, I should look for what is the most likely symbol, the maximum likelihood symbol, based on the symbols of the children. So for example, if all of my children had an A in the first position, but like most of them had a C in the second position, and then maybe almost all of them had a G in the third position, I would kind of just use those models of evolution to figure out what is the likelihood of having an A, C, G, or T in every single position of my tree, and then just take whichever one has the highest likelihood. There's a question, can I define a node again? Yeah, so in my tree structure, so yeah, that's a good point. Yeah, so in a tree, each of these leaves is a node, and each of these internal like intersections is also a node, right? Each leaf represents a present day sample, and each internal kind of intersection, which we'll call node, represents an ancestor. So basically, a post-order traversal would be, let me look at the leaves first, and I already know their sequences because that's what was given to me. And then let me look at this node and see what do I think its sequence should be. Then let me look at this node and see what do I think its sequence should be. And then let me look at, and like this one's sequence depends on its children. This one's sequence depends on its children. And then let me look at the root, which is also a node, the common ancestor of all of them, and its sequence will depend on the sequences of its children. So that's what I mean by a post-order traversal. We just kind of visit every single leaf and every single one of these internal nodes, these intersections, kind of up the tree. Yeah, great question. Any other questions about that? And yeah, basically what we're going to be doing is using those models that we talked about last week, whichever one we think is appropriate, we're just going to take it. And then for each position of my genome, I can say, given that these are the sequences of my children, what is the likelihood of seeing an A versus a C versus a G versus a T at each position of my genome? And whichever one's the most likely, that's the one that I'll say I have at that position. And the result is that I can basically label every single node of my tree using my best guess of the sequence. So every single one of these branches, I can just kind of label it with what I think the best guess is for the sequence at that node. So yeah, this might be this node sequence, this is this node sequence, and then this is this node sequence. Does that kind of make sense? So by doing this, I've now discovered not only the evolutionary tree, but I've actually predicted the evolutionary history of the specific mutations over the span of my tree. So I can say that like C at this position originated somewhere along this branch of my tree. And like this deletion here originated somewhere along this branch of my tree. So there's a question, how do the children predict the parent node? Basically, what we do is at any given node, we look at what are the sequences in each of my children. So for example, let's look at this one. So this one, maybe this child has an A, and this child also has an A. So under the models of evolution that we talked about last week, I would say, what is the probability that this first letter was an A, given that both of my children are A's? Versus what is the probability that the first letter is a C, given that both of my children have A in the first position? That's not likely, but it's still possible. It could have been that this was a C, and then both of these mutated into A. It's not very likely, but still possible. What's the probability that the first letter is a G, given that both of my children have A? So on and so forth. And then I can use those probabilities to then say whichever letter has the highest probability, that's the letter that I'm going to pick. Cool. Yeah, any other questions about this? So now we can overlay this information on top of the evolutionary tree to actually see like when exactly do we think each mutation was introduced? This mutation happened on this branch, this mutation happened on this branch, this mutation happened on this branch, and this mutation happened on this branch. I can see like which node is the first one to have that mutation. That means it must have been introduced in the branch going into that node. Cool. Okay, so the next section is called dating of phylogeny. So essentially, when we infer a maximum likelihood phylogeny, like how we talked about last week, like we plug it into IQ tree or fast tree or any of those tools that I told you about. You get your multiple sequence alignment, run it through this tool, and out comes an evolutionary tree. That tree's branch lengths are actually in terms of number of mutations. The evolutionary tree, we didn't tell it how much time passes between each of the sequences. So it doesn't have any idea, are these seconds or minutes or hours? The only information we gave it was the sequences, and as a result, mutations that occur along the tree. So the branches that we get from those phylogenetic inference tools, the length tells us how many mutations occurred from parent to child. It doesn't tell me how much time passed. It tells me how many mutations occurred. So if I'm dealing with viruses, viruses evolve pretty fast compared to, like, if I'm dealing with humans, humans don't evolve very slowly. Like, not very many mutations happen from parent to child. Right? Like, we're dealing like generations, you'll get like a couple mutation. I mean, okay, quite a few more than a couple, but either way, viruses like mutate very, very quickly comparatively, but we don't know how many mutations occurred, or like we don't know how much time, only how many mutations. What would be much more useful for us is if those branch lengths were in unit of time, because what we're trying to do is study an epidemic. We're trying to study, how did this virus evolve over time? How did it change over time during our epidemic? So what we want to be able to ask is, how much time has passed from parent to child, right? For example, if I have a bunch of COVID sequences from San Diego, and I find the ancestor of all of those COVID sequences from San Diego, the time that's passed from that ancestor to those sequences gives me a rough estimate of when was COVID introduced in this population, right? So that's how I can actually get some helpful public health information. So yeah, so the thing is, I can only infer a mutation phylogeny. So branch lengths are how many mutations happen using just the sequences. What I need to do to have information about how much time has passed is I need some information about the time at which I observed the sequences. So for example, I could have sample collection dates. So maybe I have a bunch of sequences from San Diego, and in addition to the sequence itself, whoever did the nasal swab jotted down, oh, today is November 8th, 2021 at, I don't know, 6 p.m. So we actually might have timestamps on every single one of the leaves. And what we want to do is try to use those sample times, use those timestamps that we have of when exactly was the sequence collected, to then scale the branches of our tree from unit of mutations to unit of time. Does that make sense? So the tree itself only has information about the mutations, but given that we know exactly what time each of these leaves was sampled because someone physically did a nasal swab and wrote down that time, I want to scale the tree such that those leaf times line up with what they actually were. So by scaling the branches, what I'm essentially doing is estimating mutation rates. I'm estimating how many mutations occurred per time, and then I'm scaling the branches accordingly. Right? Because if I know, like, this branch is 2 mutations from my mutation tree, and I know that like 1 mutation happens every day, then that means that it must have been 2 days, this branch. So scaling the branches is the same thing as estimating mutation rates, because then you can just convert between them. Okay. So does the kind of computational problem here kind of make sense? What we're trying to do is basically, for each of our branches, we know how many mutations occurred, and we want to overlay time information to actually estimate how much time has occurred instead. Does that kind of make sense? Okay, cool. So again, fortunately for us, the central theme of this class is that we can do stuff with the help of a bunch of these really smart people. Tools already exist to do this. So these different tools make different model assumptions of how mutations, like how mutation rates are over time. So one popular tool is called tree time. And the cool thing about this one is it can scale the tree to be time, and it can also do the ancestral state reconstruction that we talked about earlier. Another tool is called LSD2. Another tool is called log date. These are just a bunch of different tools that approach this exact problem, but they just do it in different ways. They have different techniques for trying to do this conversion from number of mutations to number of times. There's a question in the chat. How can you scale a time factor if someone collects one sample at an early time versus a different sample 1 week later at a really late time period? How can you scale time if collection times might differ? So actually, the fact that the collection times differ is important to us. If someone samples the virus a week ago versus today, well, COVID has roughly one mutation every 2 weeks, if I remember. I think so. So theoretically, if we have like maybe one sample was collected a month ago and one sample was collected today, and we see that there's 2 mutations that happened over that time period, then that tells us some information about like, oh, that can help us scale these branches such that this 2 mutation branch corresponded to a month of time difference. So actually the differences in the sample times are exactly the information that we're using to figure out how to scale them. Because we know how many mutations happened. If you also tell me how much time has passed, that tells me the conversion. Does that kind of make sense? Yeah. Cool. So these are a bunch of different tools. They all take different approaches. They each have their pros and cons. But yeah, and then you can just kind of try one out. Okay, so there's another problem called assigning genomes to lineages. Basically, a lineage, you might have heard of like Delta variant or like B117 or all these different COVID-19 lineages. A lineage is just a subtree of the overall tree. Like they sound scary because they put all these weird names on them. It's just a subtree. So biologically, it's all viral sequences that inherited all the mutations up to a certain point, right? Because if you have this subtree, you have like your whole evolutionary tree and you have like this one subtree, maybe it's Delta variant. Biologically, that just means every single sequence under this subtree has accumulated all the mutations that happened up to the ancestor of that subtree. So they all have a recent common ancestor that they've all inherited all the mutations from the old ancestor all the way down to that point. So yeah, lineage is just a subtree in the overall tree. A strain is a significant deviation from the ancestral virus. So you might have heard of like HIV strains or like influenza strains. A strain is just, you have a really, really big deviation from the ancestral virus. It's still the same species, like influenza, no matter what strain of influenza you have, it's still influenza, it's still the flu, versus like whatever strain of HIV you have, it's still HIV. It's just, it's differed large enough from the original ancestor such that they now designated a new strain. And me being a computational person, I like firm and specific definitions. And I actually could not find a firm and specific definition of what exactly they mean by significant. So in general, I implore you all, if you ever do any data analysis type of thing, always be super strict with people when they use these fuzzy words like significant or big or whatever. Because a lot of times I try to look into these things and there's not like actually a specific definition of what they mean. So as I was like going down this rabbit hole of figuring out, wait, what exactly is the criteria that says it's differed enough to be significant to be a new strain? I couldn't really find any specifics, but it seems to have something to do with any specific kind of like significant functional or phenotypic differences. So remember, phenotype is like the physical presentation of something. So it seems like it has to do with if it's mutated in a way that the physical appearance of it or the like actual functionality of it is substantially different. That's what dictates a strain. But even then, what does significant functional or phenotypic difference mean? I have no idea. So yeah, so I'm not an expert virologist. I don't know. My best guess is that they probably just have a bunch of experts sit in a meeting and just argue if they think it's significant enough. But yeah, alas. So for SARS-CoV-2, people use this tool called Pangolin to assign genomes to lineages. So if you have a new genome sequence that you've collected from San Diego and you want to know, is this Delta variant? Is this the UK variant or whatever? You can use this tool called Pangolin, which is open source, and that assigns it to a lineage. It tells you, I think this is Delta variant and it's this specific sub lineage of Delta variant, like stuff like that. All right, any questions about that? So basically all it's doing is it's saying, we already know that all of these different lineages exist. Here's this genome you're giving us. Let me just tell you which one it best fits. Okay. So what can we do with that information? They've told us, I've given pangolin my genome. It's told me, here is what lineage it falls under in the overall COVID tree. What do I do with that information? One very helpful thing that we can do that really helps with public health efforts is studying the prevalence of mutations. So what we want to be able to do is track, how have these mutations spread across the world? And the reason why this is important is because if one mutation or a few key mutations are becoming super, super prevalent, they're showing up everywhere, that might mean that those mutations are really, really, really important for the functionality of the virus. And by knowing that type of information, that can tell us how we want to design vaccines. That might tell us how do we want to design therapeutics. It basically tells us what is important for the functionality of this virus. Because if it becomes super popular, that a lot of the times will imply that it might be very important for its functionality. So it might be the thing that we want to try to break to fight the virus, if that kind of makes sense. So there's a few different tools for studying the prevalence of mutations. One tool is called NextStrain. It's kind of the most popular tool for tracking viral mutations. I've showed it a few times in this class, but basically it allows you to visualize the evolutionary tree plus the mutations that have happened, plus demographic data, like what country is it from, what day was it sampled, like what other interesting demographic information are there, like maybe the age of the patient, stuff like that. So you can find NextStrain on this website. Maybe I'll just pull it up real quick. Are you all able to see the NextStrain screen? I think, yeah, cool. Yeah, so NextStrain, you can kind of like click on it and they have this global analysis. I think I've showed it before. Where, yeah, you can kind of, hopefully now it makes a little bit more sense now that we've talked about the tools that are being used. But basically, I can say like, here's the global one. Let me color it by clade. Clade is another name for lineage. So these are all the different COVID lineages, like Delta, there's a few different types, Kappa, Epsilon, whatever. And we can see that like, oh, green is Delta. There's a lot of Delta. Versus maybe I want to color it by something else. Maybe I want to color it by, oh, here's pangolin lineage. So these are like their kind of proprietary lineage definitions. Why is it frozen? Uh-oh. Is it too intense for it? Oh no. It's frozen. Oh, there we go. So these are the pangolin lineages, whatever that means. And it also has like the globe, so you can see like what is the proportion of each of them. I think I'm gonna close out of it though, because it's like murdering my laptop and I don't want my laptop to freeze. But the link is in the slides. Feel free to like play around with it. They also have these reports. They have some data sets you can download. Pretty cool stuff. And they also, I mean, they became really popular to the general public because of COVID, but they actually originated with the flu. So you can use this to track like the flu over time, stuff like that. Cool. Let's see. How do I go back? There we go. Yeah. So that's one popular too. Yeah. Too much data. Exactly. Oh, and I had a screenshot of it. Whoops. So that would have been helpful, but that's okay. So another popular tool is called Covariance, and Covariance lets you track variants and mutations over time. So I'm going to click on it just to show you, but I also have a screenshot in the slides. But what I could do is like, let's say per country, I want to see how prevalent are the different variants. Let's say for the United States. So in California, it looks like Delta is the dominant one. It seems like, when is this? Somewhere in April, it was introduced, and then it kind of blew up. So that tells us that, hey, maybe Delta is important functionally. Versus like before that, it looks like this purple one, Epsilon, was the dominant one. But then this red one, alpha, kind of took over, and then delta took over, and then right now everything's pretty much delta. Huh, that's actually kind of scary. What the heck? I didn't realize that it was like all delta. Ooh. Okay. Well, yeah, that tells us, hey, delta might be super important because all these different states, it's like taken over completely. So then what that can help public health officials is like, hey, how can we, yeah, Delta Airlines is taking over, exactly. So yeah, so what that tells public health officials is like, if we, so like with the booster shot, I don't know why they use the exact same formula as the original one. What they could have done is change the formula to actually have some of the unique mutations that are in Delta be a part of the mRNA in the vaccine. So I have no idea why they didn't do that, but theoretically, that's one thing you could do. And with the flu, that's something that they typically try to do is they, the yearly flu shots that you get, they see what mutations are becoming prevalent elsewhere in the world. And let's design a vaccine that hopefully targets those mutations. So they're trying to kind of like predict what do we think is gonna come here, and then design a vaccine to target that specifically. And yeah, like treatments, therapeutics, stuff like that. Cool. And yeah, so this site, it's pretty cool. They have a lot of like interesting things you can play around with. Yeah. There's a question. So is the area under the curve that matters or the height? The area matters. Yeah, great question. Yeah, so if I go back to it, it's the area that matters. You can think of it like every single slice of this is kind of like a stacked bar chart where the areas, like the heights of each individual piece are important. So it adds up to 100 because you have 100% overall. But yeah, it's the area, or really like the vertical distance at any given point, that matters. Cool. Okay. And then I had a screenshot. You can do a lot of cool analyses of like different countries and stuff. And then outbreak.info is another useful repository. They track cases, deaths, lineages, and a bunch of other information. And the cool thing about this one is that they have, so I mean, first of all, they have a lot of interesting visualizations. So let's say that I want case and death data in San Diego. Why do they have Carlsbad? What the heck? That's like so specific. But yeah, let's say that I want to look at San Diego in general. They have this like pretty visualization. I can make it log scale. I can download in the visualization and the data. Yeah. Versus, let's say that I want to look at variance overall. I can click on the variance section and do the lineage mutation tracker. And it has all the different variants, and I can kind of look at them and see what mutations does it have, what is some info about it. Maybe I want to see, well, for each location, what variants are being prevalent. I can look up San Diego again, California, not Colombia. And it tells me like pretty much the same information as the other plot, right? Where we saw the prevalence of each lineage over time. Right, so this looks pretty similar to the other plot. Yeah, we can see case counts, all this useful stuff. And for anyone interested in going into more computational stuff, there's a question in the chat: should not epidemiologic fields pay attention to the rate mutations, like family doctors and stuff? I'm generally, yeah, that's a great question. So I think in general, so pre-COVID, I was very much in the boat that everyone should look at the data and kind of play around with it. During the COVID pandemic, I think a lot of people misinterpret what data mean and kind of like, I don't know. I'm a little bit more paranoid now that people don't know how to interpret the data and they don't take a class like this one to learn. So I think I prefer epidemiologists to kind of come up with general public interpretable reports. So like with NextStrain, they actually have these like reports in addition to the data. and it tries to explain it in a way that makes sense. Because yeah, like doctors don't have very much like virology background. They have more so kind of what viruses are there and what treatments do we use on those viruses. But they don't really know how to understand epidemiology. But yeah, that's a good question. So yeah, I like giving people the data, but also maybe including info that they can kind of interpret. Yeah. Okay, and then the last thing that I want to highlight about this one is that they have this API here. So in computer science terms, an API is basically just a way that you can do automated queries of some database or service. So you can actually automatically, in a very like systematic way, query the data from this website. So if you're maybe building a web app or like a Python program or like some tool that you want to code that uses data from this database, but does your own thing with it, you can do kind of like systematic queries against this database using their API. And they have a couple examples. So you can do like, I don't know, like, let's see. So I don't know what these means. Like, let's say this is something to do with Kansas City. So let me like submit it. And then it tells me like, here's the returned output. So this doesn't make much sense to us. But if you're writing a program, you can write a program to build queries like this and then parse the output. There's a question in the chat. Could you send the link again? Yeah, I can paste the link to this one, and it's also in the slides, I think. So let me just copy the link to outbreak.info, and then their API specifically is right here. But all these links are in the slides as well. Yeah, so if you're ever if you ever take a coding class, or if you ever just out of fun want to make a project, this might be a good resource to do it based off of. Cool, so I'm gonna go back to my slides. And yeah, and just here's a screenshot of like what you already saw, and kind of touching on Kevin's question, like this is what I mean, where it's nice to have data, but then having like a paragraph that just kind of describes what the data mean, to put it in context, I think is a good way of presenting it. Yeah, and that's a very, I think, important science problem of not only should you be curious about like how do you come up with answers, but you should always be thinking about like how do you communicate those answers. I think a lot of scientists focus so much on the science and not as much on the science communication, but both of them are very, very, very important.